will be also awesome. So definitely you don't want to miss those. Now here on my last lecture, I come actually back to what I started with. If you remember, long time ago, in September, we talked about oncolytic virus, and we had a simple oncolytic virus model. And then I introduced all kind of concepts of stability, linearization, Lyapunov function, and so on. So in my last personal lecture, I come back to exactly the same topic and talk about oncolytic virotherapy again. But of course, this time I want to carry it much further. And I want to use ideas which we discussed in class, and also relate this to specific experiments. Yeah. Also, what you see here is Tolo, this red character. I use him in my calculus classes. So he then helps me to explain calculus concepts to the students. So students love Tolo. Now, this is joint work with Awa Babdullah. She is now a professor in the United Arab Emirates, and she did a PhD with me. Negar Mohammad Nejad, she is a PhD student who is also currently here at the Fields Institute. Morgan van Balsom was an undergraduate student working with me over the summer, and now she is back at Queen's University in Kingston in Canada. So full of women power here. So let me talk again about oncolytic virotherapy. As I said, it is long ago that we talked about this. So the idea is crazy. So you want to use a virus to treat cancer. And typically virus is considered to be something bad and something which makes you sick and you want to avoid. But here we want to actually use it for something good. We want to use it to cure or at least inhibit the growth of cancer or control the growth of cancer. There are two approved oncolytic viruses. One is a herpes virus. It's called TVEC. It's used for melanoma. And the other one is called 85YCD mutated TKSR39-REP ADP. It is an adenovirus and is used for head and neck cancer. What I'm looking at later is called viruses of the reovirus family. I show an example here already. I mean, of course, right, you can imagine there are many different families of viruses and they look different, and there are HIV viruses and so on. There's hepatitis A, B, C, and all these kind of things. And you can classify them in families. And so later I will look at reovirus family, which is double-stranded DNA. DNA is sitting inside this virus, which is surrounded by certain layers of protein, which produce, in this case, a hexagonal kind of arrangement. Okay. So the idea is that the oncolytic virus, starting here, actually infects both. And it infects healthy cells and it infects cancer cells. But in healthy cells, the machinery is intact and the virus cannot reproduce. It is just infects the healthy cell and then it's destroyed because the healthy cell is healthy. However, in the tumor cell, it might not be destroyed because some of the repair mechanism or so might be dysfunctional. And then the virus can replicate, and then the tumor cell lyses, which means it explodes essentially and releases new virus particles. And then a cascade arises, right? So then these new virus particles can infect the next tumor cell and do the whole process again and infect and infect and infect. And so then this leads to 3 effects here. Let me just see in which order I want to look. Yeah, so this is one, maybe the first effect, a release of new virus particles infecting more tumor cells. This is exactly what we want. The second one is release of tumor antigens. So antigens are markers, right? And even depicted here. So the infected cells looks more brownish than the healthy cell, which looks like a healthy tan. So, so this is essentially the antigen representing so that the immune system can recognize those cells and kill them. So there's a systemic anti-tumor response due to this antigen. And the third effect is destruction of the tumor microenvironment through the lysis of the cells and so on. So to create a whole mess of stuff where the tumor was. Now, there's a bottleneck here in this process, and this is between the items 1 and 2. So 1 infects the tumor cells, but 2 sends in the immune response to kill the infected tumor cells. If you do that, if you kill the infected tumor cells too quickly, then they have no chance to replicate the virus, and the virus has no chance to infect the 3rd or the 4th or the 5th cell. So the infection here, this needs to be fast enough so that you can actually reach all the tumor cells before the immune system comes in to kill everything which has a virus signature. Is there something? Yeah. Update, no. This one here. Okay. Found it. Good. Right, so there's a bottleneck. You want to be fast enough infecting everything. And because the immune system will come in and clear everything out. And if you leave behind cancer cells which were not infected, then it didn't work. Okay. So the mathematical modeling of oncolytic viral therapy has a very long, not very long, but has a long recent history. So I would say maybe it started really in 2006 with papers by Wodars, Dingley, Friedman, Tian. There were some earlier papers by Helen Byrne and Dominic Wodars, but it really took off in 2006. And then in particular, Tian, he wrote like a sequence of papers of generalizations and adaptations and changes, etc. And I will go through some of the results here in my presentation. Then Ftimi and Jackson, they include the immune response, immune response into the models, which we saw just in the earlier schematic that this is important, and Pouladvant actually as well. So I need to extend my bracket to those guys as well. All right. So what I like to present in this class today is summarized in these 4 papers. The first one is with Awab Abdullah on spatial oscillations in the Bulletin of Mass Biology. Then we have a paper in revision where we fit the model to the reovirus data. And I talk about this. So this is on the bioRxiv. And then with Morgan van Walsum, we included also the immune response. And this is not published. It is just a manuscript at the moment. What I really like to recommend is the Math Oncology blog. So there's a blog site which is called Math Oncology Blogs, and I wrote a blog on the eye of the needle in oncolytic viral therapy. And this should give you a very good introduction to the field and to the interesting questions here. Okay. So let me just slip back to all the literature here, right? So this literature, of course, contains lots of mathematical models. And if you go through these, you find there's something like a base model, which essentially everybody uses and works from. And the base model is this here. You want me to go back? This one? Yeah. So the base model then would be this. And I'm not going through the detail of modeling this step by step. I really just show you the entire thing. So we have cancer cells, C, and we have infected cancer cells, which are called I, and we have virus particles, V. And the different terms here are very standard terms. So for example, we have a logistic growth for the cancer cells, and then the C plus I term indicates some competition for resources between cancer cells and infected cells. The infection happens here with the infection rate beta. If they are infected, they become infected cells, so they end up here. And they die mostly due to lysis. So after the virus has matured, then the cell dies. And upon lysis, we create new virus. And an important parameter here is this B. The B is the burst size. So this essentially tells you for each cell that dies, you get maybe 1,000 virus particles, so something like this. Then we have a virus loss due to normal washout or through the blood system, for example. And then there is a red term, which is a term virus loss due to infection. So if a virus particle interacts with a cell, then you lose a healthy cancer cell, but you also lose a virus particle. And there is a discussion. So some people say this term is very important. And other people, like myself, say this term is not relevant at all. Because virus, right? Virus particles are in the order of 10 to the 9. These are huge numbers, unbelievably huge numbers. If, let's say, 1 or maybe 10 virus particles bind to a cell, you can neglect this term. So this term kappa CV is small. In my books, this is not just small, it is very super small. So small that it can be ignored from the equations. But there are other people who say this term should be included just to have a balance of kind of forces or so happening. So small actually means the kappa, right? The kappa thing here would be small. Okay. Yeah, so this is actually the first test I want to do. So I have a base model which has two versions, one with the kappa term and one without. And I want to see if there's actually a big difference between those or not. And before I do this, I do a non-dimensionalization. This is something I discussed, I think, in the second week in September, where you just reorganize your parameters, make them non-dimensional, you define ratios of certain parameters. And then you get this system where you only have the parameter A and theta and gamma, and this mysterious kappa to worry about. So we are down to 4, and we started here with 1, 2, 3, 4, 5, 6, 7, and we started with 7. So it's quite powerful. These models are equivalent, so whatever we prove for the non-dimensional system is also true for the dimensional system here. Okay. And then one parameter I want to single out, which I call theta, which is a combination of the alphas and betas and so on, and in particular of the B. Remember, B was the burst size. So how many new virus particles you get from a bursting cell. And so then I call theta kind of the effective burst size. This is essentially a burst size parameter here. And this will be the bifurcation parameter. So I will analyze what happens if theta is small or big. Well, if theta is small, you don't get many viruses per infection. So then the infection cycle will probably break down. If theta is larger, you might be successful to infect everything. And so it's worthwhile to look at this. And I think this is here. So here's a bifurcation diagram. Yeah. So a bifurcation diagram, you vary a parameter. Here I actually wrote B, but I mean theta. In my mind, they are interchangeably. So I vary this parameter theta. And then on the y-axis here, I plot the corresponding steady states of the system. Okay. And this is only for C. So this is a C, let's call this C bar steady state. So if theta is small, maybe somewhere here, then the steady state is 1. So cancer wins. Okay, then cancer is a carrying capacity is 1. So up here, cancer wins and the virus dies out. But if I make theta a little bit larger, so maybe past the value here of, let's say, 6 or so, or 5 point something, then you get a bifurcation, and this is a transcritical bifurcation. Transcritical, if you want to look that up, or maybe even know. Happening here, where you get a coexistence steady state here. A coexistence steady state. Not good to read, so this could be anything. Coexistence steady state, where you have cancer cells, infected cells, and virus particles all together. And now you see two curves. You see a blue and the red. And so the blue one is kappa is non-zero. And the red one is kappa is 0. Okay. And if you look at these pictures, there is a difference. The value for the bifurcation is shifted a little bit, but the overall qualitative figure is exactly the same, which is nice. Good. And so then red and blue come closer together here in this area, here. And this branch leads to oscillations. So we only show the maximum and the minimum value of the solution, and the solution really oscillates between the up and the down and the up and the down. So this is how you should imagine this. I take this out of the picture again because it doesn't look good. But that's what's happening here. So then this is a Hopf bifurcation. Hopf bifurcation right at this point where you get the periodic orbits coming out. So this is the bifurcation structure of the base model, and this has far-reaching consequences for everything else we do. So for low theta, cancer wins. For intermediate theta, cancer is reduced, but it's still there. For high theta, we run into oscillations. Now I plot my oscillations again. But every now and then we reach very low levels of C, low levels of cancer C. And this gives us hope. So maybe at those points, actually, we can control the cancer, or maybe it goes extinct just on its own because we reduce it so much. So the good case is the oscillatory case. This is where we want to be with our model. Okay, so here I have the solutions as functions of time. I used the parameter values by Pouladvant et al. From their paper. And this gives me a base value of theta is 58.33. And in this parameterization, this is one of the coexistence steady states. So this is right here where we have coexistence. And now, if you plot this as functions of time, you see some initial going up and down, but then everybody settles here on a steady state. So C is red, cancer cells in red, infected cancer cells in black, and virus particles in blue. And the virus is essentially close to zero. So cancer persists. If I increase theta, okay, I get closer to the bifurcation point, but I'm not past it. Then I get oscillatory behavior first, but then again, it settles on a steady state. And then if I get past the Hopf bifurcation point, the Hopf bifurcation point in this parameter is 338. I can compute this. So if I get past the Hopf bifurcation point, I get oscillations, and you see them as these spikes here. So red is cancer cells, black is infected cells, D is a virus. So cancer grows up, and then the virus comes, infects everything, and everything goes down again. Okay, so that's the oscillatory case. Now, again here, we see that between these oscillation peaks, we get very low concentrations. So C low. And between all of these. So we would like to know if this is enough to treat the cancer or not. And this can be done by the tumor control probability, which is here. Oh yeah, and I have the formula here. So tumor control probability is a probability that a cancer is eradicated. And in its simplest form, this is the Poissonian form. So there are different models how to do that, but the simplest one is the Poissonian one. It is e to the minus C0, which is the initial condition, initial cancer size, initial condition, times surviving fraction. S of t is a surviving fraction. Okay? And the surviving fraction you can compute from these graphs, right? So here where I put my red arrow, I know what the solution for the red curve is. I know the initial condition. I can just compute the ratio, and I know the surviving fraction. So we compute the surviving fraction from the pictures upstairs, plug it into this e to the minus thing, and we get the probability of cure. So probability of cure means you want to be close to 1, right? So, and then here, so you see here we get close to 1 at the beginning. Here we get close to 1 between the oscillations, and here as well if theta is higher. So this is where we like to be. And I consider this this case here like is a transient case. It is not, and it goes down and then it settles about 40% here. Whereas in the oscillatory case, we get close to 1 every time we come back. And this is where we would like to be for treatment success. So now let's include space. Now we come into the realm of reaction diffusion equations. So we include a diffusion term for the cancer cells, for the infected cells, for the virus. They have all different diffusion coefficients. In particular, the virus is much smaller, so it would diffuse much faster. And now the base model here is oscillatory. So then the question arises, what happens if I couple these oscillators? Okay, let me draw a one-dimensional picture first. So let's say we have a domain here, and here are the cells, and they're all lined up. I do a one-dimensional picture first. Give them a little nucleus. Okay. Now, so let's call this 0 to L. Now, each of these systems here is oscillatory. So this one will oscillate, this one will oscillate, this one will oscillate, the neighboring will, but then they're also coupled. They're coupled to the diffusion term. So then the question is, what happens to the oscillations? Will they synchronize? Will they oscillate at the same speed and the same rhythm? Or will this be, I don't know, very chaotic or so? So the question is, how do these coupled oscillators behave? Well, they oscillate in the Hopf region. They do not oscillate if theta is small, okay? So if theta is small, then this system is actually boring. But if theta is large, this is non-boring. So here we go. Again, two cases, theta is small, theta is big. Here I show the cancer cell density in brownish colors, the infected cells in blue, and the virus in blue to red. And we infect, let's do this here, we infect the cancer with the virus in the middle of the domain, and then time goes downwards. And then we see how the virus spreads. Oh yeah, how the virus spreads as time evolves over the entire domain. And then after the virus has infected everything, we get a homogeneous steady state right here. And for the infected, we get a light blue level here. And for the cancer cells, we get brown, a homogeneous brown here. So again, we have one infection cycle, and then we settle on a steady state. It's different for theta is big. We also get the invasion of the virus like this and this, but then it repeats, right? It repeats and repeats, and then eventually we get a periodic solution. And if you run it longer, it becomes completely flat. So here the periods synchronize. They all go together, like a heartbeat. Okay. Yeah, now in 2 dimensions, no, not in 2 dimensions. Here we use 5 inoculations. Earlier we inoculated in the middle. Now we do this at 5 locations, 1, 2, 3, here are 2, 4, 5. And you also get the spread for small theta. For larger theta, you get periodic solutions as before, and they synchronize very nicely. Okay, so how does this look like in 2D? In 2D, then, we get what we should expect is like a ring. So here we infect with the virus in the middle of the domain, and then you see a ring spreading out. Oh yeah, here T is 1, here T is 3. Okay, so starting initially, and then it spreads out like a ring and infects the cancer cells. So here are the infected cancer cells in the middle, and it's spreading out. So this is exactly what we like to see. Now this is oscillatory, so it should repeat if I run it longer, and it does. So we get a first ring by time 3, and then time 13, it starts again. Time 16 looks similar to time 3. 26 looks similar, and 38 looks similar. So it repeats. It's a pulsating thing, and the ring spreads out, but it doesn't seem to synchronize. There's also the effect of peak splitting. So here we get 2 peaks coming out, and here as well. And we were not sure what that is. So I've never seen this effect before. I think there's some form of bifurcation sitting underneath, but I have no idea what that is. So if somebody wants to analyze this peak splitting, it's a interesting mathematical problem. Probably not easy. Okay. So, but now if you run this longer, you get actually chaotic dynamics. So here we use 5 inoculations. And you see these concentric rings spreading out. But then if you increase time, this becomes quite chaotic. And if you run time much longer, you get very chaotic patterns. And this was very unexpected. So there are spiral waves, there are target patterns, there are like all kind of stuff. And actually, since we are among us and we have a little bit of time, I show you these simulations using Visual PDE. So Visual PDE is a wonderful software which is available online, where you just can enter a PDE and a few parameters and you can run it. And I'll show you how this looks. So where's my Visual PDE thing here? So here you see on the top left, you see the Visual PDE thing. Let me do a reset and reset. Okay. And so you see I entered the equations here. I have the three equations here. It's called U, V, and W. And this is our virus, oncolytic virus model. And then when I tap it with my pen, I can make initial conditions for the virus. So let's tap it here and maybe here. So now I have 3 inoculations. And then I can let it run. And then you see it spreads nicely over the domain. Actually, I move this away. Okay. And they come back. So the periods are definitely here. It's kind of mesmerizing. It looks really good. And then I just let it run a while and have a sip of water. Yeah, it looks already very different. It found kind of a different period, a different frequency. Some waves are moving out, some waves are moving in, as it seems. And now in the middle, it gets already very chaotic, and this will then spread further to the outside. I could watch this for hours. Oh, I see. It's a little bit rugged in the projection. So it's much smoother on the computer I have here. But there you go. It's like looking at the clouds. You see patterns, you see animals, you see all kinds of things. And then this kind of is now relative persistent. So there seems to be a dominating frequency steering this. And so this is what you get. Yeah, so if you want to play with Visual PDE, I can recommend this. This is fun. Okay. And this is also then laid out in the Bulletin Math Biolo paper with AWA. Okay. So now let's come to the Rio virus. So in Edmonton, we have a lab by Maya Shmulewicz in the Li Ka-sheng Virology Institute. And she asked me if we can help her to analyze some experiments and some data. And the idea is to create a reovirus, which is highly efficient as an oncolytic virus. And I don't know if I told this story in this class, did I? Where I went to see Maya's lab. You did. I did, right? Yeah. And then she showed me the virus. Yeah. So, and then he showed me the virus in a petri dish just under my nose, and I was surprised. Yeah. Okay. So this is what she showed me. And so what you see here in blue color are cancer cells, breast cancer cells. And then they have been infected by a virus, and every infected area then becomes white. So then in particular here in the middle diagram, I can make it even bigger, we see a lot of white dots, and these are called the plaques. And you can then measure all kind of things. You can measure how long it takes to form them. You can measure how many you have. You can measure the area, how large they are. And this then gives you an idea about how infectious the virus is and how long it takes to get maybe a more complete infection like over here. Okay. But I didn't present all of this to class yet, right? I didn't. Okay. So yeah. Oh yeah. Now let's learn a few facts about the Rio virus. So it is a double-stranded DNA virus. Other viruses might be RNA viruses. Okay, so this has actually DNA. There's a wild type, which is called T3WT, and this resides in the digestive tract of mammals. So probably all of you will have this virus in their guts. So, which means it is not pathogenic to humans. It just helps with the digestion. But it has anti-tumor abilities and is now considered in over 30 clinical trials for all kinds of cancer, for metastatic breast cancer, for prostate, for colorectal cancer, etc. And in particular, two phase 3 trials are happening, and phase 3 means it is very close to becoming a medication, right? So now it's really tested on patients to see how they react for breast cancer. Okay. However, a preliminary evaluation of the trials, only 21% of the patients respond to the treatment, and this is not enough. We want close to 90% of patients to respond. And the question is, how can we enhance the oncolytic activity of the reovirus? And so this is a question the Smulewitz lab had, and they systematically developed mutations of the reovirus to increase their oncolytic potential. And among these many mutations, they found one, which they call supervirus 5, which has 5 mutations. And this seems to be producing larger plaques. So meaning it infects more cancer cells than the wild type. However, the binding rate of the supervirus is lower. And this was a big question mark because people thought if you have a virus which binds better, it should infect more cells. But here we see the opposite. We have a virus which binds less, but still infects more cells. And the virologists were really confused about this. And it was one reason why they approached us, because they saw like a little paradox. So lower binding rate and getting larger plaques didn't really make sense. So I think
we are asking the virus too much.
maybe we asked the virus too much. Yeah, right.
Usually the virus is negative. We try here to ask the virus something good, but via infection, which is not, it may be tipping the balance of health, because infection is conducive to cancer via inflammation later.
Yeah, yeah, this is a good idea, but the answer is different, as I will show you in a second. So the question is there an optimal binding rate? So first of all, is there like a non-monotonic behavior in the binding? And then if there is there like an optimal binding rate where you can get the optimal spread? So that's what we would like to do. The data we have, they actually did a whole bunch of different measurements and data, which is really nice. But here's one set of data for the wild type and for the supervirus on days 2, 3, and 4. And then you see the spread really on a level of individual cells. And you can color them with the fluorescent protein or so. And then you can really count essentially how many cells are infected and how large the plaque is, etc. So very nice data. Okay. And so I saw these data and I thought, hey, we have this wonderful oncolytic virus models, which makes chaotic patterns and spirals and all that. So let's fit it to this data. And we tried and we completely failed. This did not work. And the reason is that our model was way too complicated. It was way too complicated for those data. So then we changed our strategy and we first looked at the data, which actually is usually a good idea, and then try to find the minimal model that is needed to describe those data. And this led us to the path to look at 2 timescales, a short timescale and a longer timescale. And the short timescale is less than 16 hours. Because a virus infection cycle takes about 16 hours. So, so in this short time, the virus can only diffuse, which is here, and it can bind to a cell, which is there, and that's it. It will not have time to produce new virus particles and lead to lysis and infects the next generation of cells. So in a short time, we have only diffusion in the medium, and we have binding to cells, and that's it. And so we do a very simple model, a diffusion binding model for the virus. One equation, one reaction diffusion equation. Okay. So we have two parameters to find, a diffusion coefficient and a binding rate. And for the binding rate, a virus was inoculated for 1 hour and then washed out from the cells. So then we know how many virus particles bind per hour. And from this, we can estimate the binding rate for the wild type to be 61.6 per hour, for the supervirus to be 24.1 per hour. So significantly lower for the supervirus. For the diffusion coefficient, they made an extra experiment for us where they took like a little rod of material, no cells is just the material the cells live in, and then they inoculate the virus here on the left, and then they divide this rod into compartments 1, 2, 3, 4, 5, 6, 7, 8, and they measure the virus expression in each of these subintervals. And then this is a curve showing here. So this is a very simple diffusion problem. This is a problem you show to your third year PDE class, and there are methods to solve this. You can use the normal distribution, the fundamental solution to solve this, etc. Do I have the fits here? No, I don't have the fits here. So, but then what you do is you kind of fit a curve through these using the fundamental solution. Maybe I write this. Fundamental solution of the heat equation, of the diffusion equation, which would be Vt, let's keep V. Vt is d Vxx. So that's the problem you solve. The solutions are known, and from that you can find your d. And here are the values which we find. So our model fit is this, 0.01 plus minus 0.0015 in millimeters squared per hour. There are also other ways to find the virus diffusion. One is the Stokes-Einstein relation. I don't have this here on the slides, but it is an old relation Einstein came up with. If you have a particle of a certain radius in a medium of a certain viscosity at a certain temperature, then you can compute the diffusion coefficient. So this is totally physicsy based. And if you use the physics approach, you get 0.02, so which is very, very close to our fit. Other people estimated this too. Rioja found a value of 0.014 and Pouladvant a value of 0.01. Again, very, very close to what we have, which is absolutely nice. So then somehow we kind of reconfirm the values which were used earlier. Okay. So now we have this guy and this guy. And we can estimate the spread rate of the virus. Let's see, what do I have here? Okay, I need to explain this a bit more. So this is a one-dimensional problem. Yeah. Come on, give me a pen. Why does it not want to write? No, it writes. Okay. Okay, so let's say what do I want? Yeah, we put in the virus here. We put in a high virus concentration at time 0. So I make this like a delta distribution, time 0. And now I make different times at different colors. So what now happens is we get diffusion, so it spreads out, and we get binding, which means we lose free virus. So then at the next time step, you maybe get something like this. Let's call this T as 1. And then at the next time step, we get maybe something like this. T is 2. And then at the last maybe time step, black, maybe we get something very widely spread out. So maybe this is T is 10. Of course, I'm making up the times, but I want to show you what happens, right? Okay. And so at some point, we lose all the virus. And losing means it is below a detection threshold. So I need to define a detection threshold, and below that, I consider the virus to be gone. Now, this is maybe a bit here. Let's just try to plot this in light blue. So I want to indicate a detection threshold, maybe this one, And the detection threshold, yeah, and below the detection threshold, I consider the virus concentration to be 0. Now, these curves, which I plotted in red, blue, and green, they have intersection point with the detection threshold, right? So here, the green one intersects here. If I go further to the left, I'm below the detection threshold, or here is another point, and this should be symmetric in plus or minus. For the blue curve, right, I have the points earlier, so I hit the detection radius here and here. And for the red curve, I hit it, well, super close here and here. So if you follow these highlighted points over time, they spread out. And this is what you can consider to be the spread radius. So then let me go black to blue. So this would be R of t, the spread radius. So this is the radius after which it is no longer detectable. And this radius spreads out until we get to the black curve, and then everything is below. And yeah, and then the spread radius becomes 0. Is this what I want? Yeah, this is what I want. And okay, and here we can compute this. So this is again a linear, non-homogeneous, linear non-hom diffusion equation. If you go back to your third-year PDE course, you learn how to solve this. And then we can compute the radius R of t as a function of time. And that is this formula right here. So V min is the detection threshold, which I just introduced in light blue. V0 is the initial condition. dV is the diffusion coefficient. T is time. And ta-da, da, 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 gamma is our binding rate. Okay, now, initially the question was, what does the binding rate have to do with the virus spread? And the supervirus has a lower binding rate, and it spreads better. So we plot this curve now as a function of the binding rate. We want to see for a given time, how does the spread radius change if I change the binding rate? And you see it's a decreasing function. Okay, so a higher binding rate means lower radius. Higher binding rate means lower radius, and this is exactly the paradox they found in their experiments. So now here blue is T3WT, the wild type, and supervirus 5 is the red one. And if you look here at the spread radii, the supervirus should spread should have larger plugs than the wild type. And this was unexpected for the biologist, and it could already be explained by a very simple model, actually. So we didn't need our full virus model for this. So the reason is then, if the virus binds too quickly, it binds to the first cell and stays there, and it doesn't spread anymore. So you need to allow the virus to actually have time to spread before it binds to another cell. That's what's happening. Good. Okay. Now we look at the longer time. Let me switch back to red. So here we want to have T bigger than 16 hours. These virus experiments are typically run on the scale from up to 5 days, 4 to 5 days. Okay. And then we get several cycles of infection. So we get infected cells, and the infected cells burst, and they produce new virus, and then the virus infects another level of cells. And here we found we got away with 2 equations. So we have the infected cells, and we have the virus. And we could assume that the cancer cells are actually more or less constant. So on this time scale, we didn't lose too many cancer cells. So we just consider this to be a reservoir. So C is constant. Okay. And let's just see how far we get with this type of model. So then we have a model between I and V. And these parameters we know gamma B, we know dV from previously. So the new parameters are then nu, A, and theta. I think I like the light blue. So nu then is the percentage of virus binding that leads to an infection. Okay, not every virus that binds to a cell actually leads to an infection. So we have a percentage here. A would be the death rate of infected cells, and theta is the all-important burst size, the parameter I used earlier already. Okay. So then 3 new parameters. So Shmulevitz, they published in an earlier paper nu to be 0.01. So only 1 in 100 virus particles leads to an infection of the cell, even if they bind to the cell. And then for the death rate and the burst size, we have extra experiments. So here, this is for the death rate of infected cells. And then here is number of cell deaths. And you see at 15 days or so, all cells are alive. At 18 days, there's a big jump and cells start to die. And then at 30 days, there's another jump, which another set of cells die. So we fit this with a logistic function. Logistic. And we can estimate the death rate, which is at the beginning here between 15 and 18. We can then estimate the death rate to be 0.057, again, per hour. And what is this? The burst size. The burst size measures how many virus particles you get if the cell bursts. And here we also fit a logistic curve. And then this level is the burst size. So the saturation level of this curve. And then here are values for the burst sizes. Here is interesting, if you look at this closer, that the confidence intervals overlap. So 514 plus 114 and 732 minus 146, they overlap. And this means statistically, there's no difference in the burst sizes. Okay, so we get different mean values, but since the confidence intervals overlap, we cannot be sure they are different. All right, so now we have all the parameters, which is nice. So then we do a traveling wave analysis. Traveling waves we talked about earlier gives you an impression or an idea of how quickly the tumor grows in a certain environment. So we do that here. This is the same model as I've shown you just earlier. And then we look for traveling wave coordinates x minus ct. And we assume there's an exponential decay e to the minus lambda in the traveling wave coordinate. And we also assume that we are at the leading edge, so the epsilons are small. So lots of assumptions go in there. But I think I did a little bit on traveling wave. So we did a little bit on traveling wave, how to analyze this. So I skip the details. I show you the results. For the wild type, we get 0.044 millimeter per hour. And for the supervirus, we get 0.059 millimeter per hour. So the supervirus is faster, right? It binds lower at a lower rate. As a result, it is actually the fastest spreader, making larger plaques, which you can see here in the red and the purple diagrams. Oh yeah, and the decay rate, lambda was the decay rate at the front of the wave, so it is actually here, let's say, e to the minus lambda z is 6.9 per millimeter and 5.4 per millimeter. I highlight this because these numbers will play a role later. Okay, so we have a model, and we fitted all parameters from the data, which is nice. And the next step is a validation. And the validation, you should use data which you did not use for the fit. You take now the model which you have, you go to another set of data and see if you can replicate the new data. And this is these data which I've shown you just earlier, where we have fluorescent images of infected cells surrounding the inoculation point. And if you run it through a scanner, you get these blue data points. So depending on the radius, you get the light intensity in these experiments. And here we can fit an exponentially decaying function, e to the minus lambda x, e to the minus lambda x for the supervirus and for the wild type. And this is the same lambda we had earlier for the traveling wave analysis. And so here we measure 9.6 per millimeter. Earlier we got 6.9. Here we get 4.9 from the blue graph, and we had 5.4 earlier. And this fits amazingly well. I mean, this is biology and not physics. So we are not looking at two decimals after the point. So 4.9, 5.4 is the same value in terms of biology. And 9.8 and 6.9 is still very, very close. So we were very happy with this result. Okay. All right. So I talked a lot about models and short and fast times and parameter values. And we probably all forgot what we want to do. So what? What did we do? What do we want? What do we want to learn? Oh, yeah. Is there an optimal binding rate? That was the question. Okay. So to find the optimum, we look at the invasion speed again. Well, the invasion speed is the thing we want to maximize. We want the entire virus infection to spread as far as possible. And now we can plot this invasion speed as a function of the binding rate down here. So the speed is here. And now let's look at one of them. Let's look at the red one, which is the wild type. So on this red curve, you see how the speed changes as the binding rate changes. And there's a clear maximum, which is indicated by the black dot. The value for the wild type is actually the red point. This is the binding rate for the wild type. And you see it is not at the optimum, right? It is a little bit too much. So you want to reduce binding rate to get to the black point here, which would be the optimal value. For the supervirus, it is the purple line here on top. And the parameter value for the supervirus is here. That's the binding rate. And the optimal value is close, a little bit to the right. So you want to actually increase the binding on the supervirus just a little bit to get the optimal spread rate. And then the blue in the middle is an average of the two. Because, as I said, we cannot distinguish the burst size between those two. And we might have to look at the average, which is the blue. And again, the maximum is indicated as a black dot, which is very close to the other values anyways. Yeah, so this is nice. So then we ran a few more test cases because it turns out the burst size and this maximum value are correlated. So now I show you a table here where we look at the burst size going from 500 to 1,000. We compute the maximum binding rate corresponding to the burst size. And then we compute the block size for these cases as well. Okay, and now this table is something we could give to the experimentalists. And they can run their experiments and they can measure this. So maybe they get 743. So maybe we're somewhere here. And we get maybe a binding rate of 0.41. So then they can kind of estimate, are we close to the optimum or not? So this is exactly what they use in the lab. So this is nice. So we are able to suggest new experiments. Another thing is this thing here. We found if we build the ratio of the maximum binding rate and the burst size, gamma B max over theta, we get something relatively constant, 56, 58, 60, 55, 56, 57, 59. So this could be another guideline actually to look at a ratio, and this ratio should be close to maybe 57 or so. Then we have a good result. Good, so this is nice. Yeah, that's what I said. All right. So then the last bit. So Morgan van Walsum, she came to work on an undergraduate project with me over the last summer. And then Nigar Mohammad-Nejad helped me to supervise Morgan. And here we wanted to include the immune response into the base model and just see what happens. And here's a way to do that. Make this a little bigger. So the base model is in black. That's the same base model I started with just many slides earlier. And the red additions are the additions to the immune response. And we have two immune responses to lymphocytes, leukocytes. So then this would be cell specific. So that's the immune response, which is directly attacking cancer cells. And then the other one is virus specific, which attacks the virus infection, which means it attacks the infected cells and the virus directly. Okay, so then the LVs show up here, they attack the infected cells, and here they attack the virus. And the cell-specific attacked both cell types X and Y. So this is a very general setup of an immune response model for oncolytic virus. If we simulate this, so now this is again an ODE, I did not use space so far. We keep the oscillations like we had before, and the new graphs are this is the LX, so the immune response towards the cancer cells, and this is LV. And it is actually the opposite. Somebody changed colors. So then green is the virus immune response, and purple is the cell immune response. So they participate in the oscillation, but they do not change them really much. We get the same oscillative picture as before. Nice. And now we look at therapies. So Morgan looked at 7 therapies together with oncolytic viral therapy. Because as we know, as I said earlier, there are 21%, in 21% of the cases it works, which means in 80% it does not work. But maybe we can boost it by using combination with other treatments. Yeah, okay. And so we use 2 chemotherapy treatments, a continuous one and a fractionated one. We use look at 2 radiation treatments, brachytherapy and external beam radiotherapy. And then we look at 3 immune therapies, immune suppression, immune boost, and CAR T-cell immunotherapy. So this sounds really scary and a lot, but for all these therapies, there are existing models already in the literature. So you can go through the literature, that's what Morgan did, found the corresponding model, and wrote it down. So why not? Okay. So then here would be continuous infusion chemotherapy. I will not now, so now we get like millions of different models here. You see, I go through very quickly. I will not show you all model in detail. I'll show you a few details. For example, with the continuous infusion chemotherapy. So then the new terms are these red terms here. And this is a very standard way to model chemotherapy. So first of all, you have a chemotherapy agent, which I call C, which then decays exponentially with the rate gamma C. So it's introduced and then it decays and then travels through the body. And then the interaction terms are typically modeled in this form 1 minus e to the minus mu I C. So that's a very, very common form to put chemotherapy in these models. So we use it. We use what other people use. And then we get this model here. Okay. If you have fractionated infusion of chemotherapy, these terms are the same, but the only equation is different is the one for the chemotherapy agent, because now we give it, let's say, once per day or so. Okay. Brachytherapy is a radiation treatment schedule, so we have radiation terms here, and then we have a radiation dose here. Fractionated radiation, same thing. We get oh, there were the radiation terms. Oh, the radiation terms here happen only at radiation times. So we stop the simulation, we radiate, and then we start the simulation again. Okay. Yeah, immunosuppression. So we mess with the immune equations here, having a suppressant, or immune boost. So we increase the efficiency of the immune system here and here. Really, I'm not going to go through the details. It's a lot. And the principle is clear, right? You modify parts of the equation and you see what you get. Okay. So for all of these now, we look at the tumor control probability. I said to you earlier that this is really the interesting bit. We want to know if you get the TCP close to 1. All right. And then for each of these seven cases, we do this here. We look at two cases. If theta is below the Hopf point, then we have coexistence of cancer and infected cancer cells, which is this state here. So oncolytic viral therapy alone is not successful. And in the middle, I show the alternative treatment alone, which now is the brachytherapy. And in purple, we have the tumor control probability, and we want this to be close to 1. So this happens here, which means the alternative treatment alone, in this case, would be successful. And now we combine them, and then the combination is also successful. It goes to 1 a little bit quicker than before, so we gain a little bit through the oncolytic viral therapy. Okay. And now I want to keep track of all the 7 treatments of all these combinations. So I think I do this here. Yeah. So I use an X to indicate it is not successful, and I use a check mark to indicate it is successful. And then I get this following table, I think. Where's my table? I first want to find the table. Here's the table. Okay. So if the oncolytic viral therapy is insufficient on its own, we get X's here all across. And then we look at how the alternative change the outcome. And it turns out that if the alternative alone is successful, then the combination is successful too. But we never gain something to the combination. So if oncolytic viral therapy alone doesn't do it, you cannot, I mean, and if you have another treatment which also is not good enough on its own, then the combination is also not good enough. That's a minor. Okay, so that's really interesting. So we do not really gain much. But also in this case, why use oncolytic viral therapy at all? We could just use the alternative treatment and be done. All right. And then for the other case here, this is past the Hopf point. So this is the point where we have oscillations. And I said this is a good case because the TCP can be raised. Here it actually at 50%. Oncolytic viral therapy alone. Here we get kind of success. This goes up to 1. And here we have success. So here the combination does a lot. It improves the treatment quite a bit. And so then my table is this one. So oncolytic viral therapy alone is kind of 50% success. But if I combine it with these treatments, I might get really, really good successes coming out in this case. So meaning essential is that we get this burst size parameter beyond the Hopf bifurcation point so that we can get to the oscillatory behavior. Yeah, so I think that's good. So, yeah, this is what I said. Good. So this is coming to the end of this little presentation. So let me just summarize. First of all, the spatial model was super interesting because it shows interesting spatial temporal dynamics. And if you want to play around with a visual PDE, I recommend you to do that. It's fun. And now I have a master student, Tejas Bansort, who is looking at the underlying bifurcations. Right, I talked to you about the peak splitting, which happens there. So there must be bifurcation there. There are also spiral waves, so we can look at spiral wave bifurcations, etc. So he's looking into this. With Nagar, we look at how these patterns change if the immune response is included. And she had shown me last week new simulations. And it turns out the oscillations disappear immediately. As soon as the immune system is part of it, you get rid of these complicated chaotic oscillations, which is very interesting in any ways. For the reovirus, we found conditions for the optimal binding rate, which is nice. And they are now used in new experiments to find even a super duper virus. And again, with NEGA, we want to do a similar analysis for reovirus and liver cancer. So we have new data on liver cancer. Earlier it was on breast cancer, so now we want to go to liver cancer and do a similar analysis there. And then combination therapies, this was the last part. Oncolytic virotherapy alone is not successful. It doesn't seem to be improved by any combinations, but also we need a more careful parameterization. So we just ran some tests. We didn't do a careful parameterization of these cases. And there's more work to be done as well. Yeah. Oh, and then with Morgan van Walsum now we want to look at oncolytic virus resistance, which is also an interesting bit here. Nice. So I think this essentially ends my presentation. Here's the references again. Again, I highlight my blog. I like blogs, so they're fun to read. Okay, so before I invite questions, a little bit administration. So so 3 of you should be working on their reports, right? And the reports are due on December 6th, I think. If you have questions, I'm happy to meet you and talk. I'm also happy to look at a pre-version, so maybe a week earlier you can send me a version. I look it over, I give you feedback, and make sure we get a good result at the end. Good, so then I end here for today. Any questions?
I have as usual, but maybe other people want to.
Yeah, there's a clap. Thank you very much. Questions? Yeah, go ahead, Itama.
Okay, I have a headache, so excuse me, but the very beginning, where the virus was attacking, and you had maybe the first slide or second slide
Right.
of the virus. I wonder if one can follow all kinds of feedbacks situations
Mm-hmm.
and explain some stuff. What's the role of the infection? What's the role of the immune system? Because the whole context here is a little bit unusual. It's a little bit virus, as you say,
Mm-hmm.
intuitively is negative. Now we try to harness the virus to fight the cancer cells.
Yeah, to use something good for
So,
us. Yeah.
so maybe, and I want to connect this to another question. These oscillations in the bifurcation region, how can we explain them in terms of this picture?
Yeah.
So
that's
you
question.
get oscillation as a mathematical analysis, and it's easy to forget why are we doing it.
Isn't it? Yeah. I had the same feeling even as I was talking. I talked so much about the mass that at some point I forgot what am I doing here. But so the second question first, the oscillations is easy explained because it's a predator-prey model. So the structure is a predator-prey, where the virus is a predator and it feeds on the prey, which are the cells. And so in that context, these oscillations are very well known and very well observed. Yeah. Your other question to look at the feedback mechanism is a very good idea as well. So there's lots of signaling going on. There are interleukins and interference, particularly with the viral infection, which then stimulate the immune response even further. So this is, I think, a very good idea. We don't have
many
data.
steps here,
Yeah.
every step here.
In every step, right? You could have
I
feedback.
can do engineering feedback
Yeah.
loops.
Yeah. And we have seen those in Dominic's talk yesterday or 2 days ago. Absolutely. I think it's a great idea. For the reovirus, the Shmulevitz lab, we don't have any data on these feedback mechanisms. But I think in vivo, these are important. Yeah. Yeah, I agree. There's a question. Okay, go ahead. Or can you read this? Has oncolytic virotherapy been applied to lung cancer? No, not that I know of. There are only two cases, which is melanoma and head and neck. Yeah, so I don't think it has been used for lung cancer. Other questions? Okay. So make sure you're all here on Tuesday for Tom Yankilov. Will be a blast. He's full of energy. He's good.
We heard him yesterday or the day before.
He was at the conference, right? 2 days ago. Yeah, exactly. Yeah.
Okay, thank you very much.
Thank you very much.
Okay, running to another thing. I'm running to another.
You are running to another thing?
Medical computer science hero is giving a talk now.
And where?
If you want to join me, I will guide you. I'll wait for you.
No, I need to meet Alan and Maria. Yeah,
So it's room 30. It's an next building.
Oh, I see.
Room
Okay.
3200.
Like in physics, I guess, right? Yeah.
No, even before physics.
Ah,
The big engineer.
the big one here.
Next
Yeah,
door.
yeah, next door.
There is coffee and some stuff.
I see. Enjoy.